Alright everyone, today we're going to be talking about the monohybrid and dihybrid process as well as the laws of segregation and independent assortments. So let's get started.
First off, what is a monohybrid cross? A monohybrid cross is when two parents that have different traits for one trait are crossed together. For example, if you have a homozygous dominant tall man and a heterozygous tall woman, what will be the height of the offspring? First, we're gonna need to draw a Punnett square, just like this one. Let's see, we have the tall man, homozygous dominant, and the woman, and the Punnett squares. The genotypic ratio is 50% homozygous dominant, which are this one on top, and 50% heterozygous, which are at the bottom. The phenotype ratio, however, is 100% tall, because having just one tall allele will make a person a tall person. Also, each square is 25% of the offspring.
And what about the dihybrid cross? A dihybrid cross is when two parents with different traits for two different characteristics are crossed together. An example would be the Mendel P experiment, the father of genetics. Mendel took a pair of contradicting traits together for crossing, for example, color and shape of seeds at a time. He picked the wrinkled green seeds and round yellow seeds and crossed them. He obtained only round yellow seeds in the F1 generation. This indicated that round shape and yellow color of seeds are dominant in nature.
Now let's draw a Punnett square for this. This Punnett square is gonna be different. It will be 4 by 4. Y for yellow seed color and capital Y for yellow seed color and lowercase for green seed color. Capital R for round shaped seeds and lowercase r for wrinkled seeds. One like this. This is the hybrid cross Punnett square with two traits at top and two traits at the bottom. Thus, the parental genotype will be a capital Y and capital R, yellow round seeds, and lowercase YY and lowercase RR, green wrinkle seeds. There were wrinkle yellow, wrinkle green seeds, and round green in the phenotype ratio, which is of 9:3 to 3 and to 1.
Now let's talk about the laws that govern these crosses. The laws of segregation and independent assortment. The law of segregation states that during gamete formation, alleles separate from each other so that only one allele appears in any given gamete, and thus an organism only has one gene governing a particular characteristic or trait at any given time.
The law of independent assortment states that the genes for different characteristics can assort independently from each other during meiosis, meaning they don't influence each other's behavior or expression within an individual organism. For example, if you have a tall plant, capital TT, and a short plant, lowercase tt, then their offspring could potentially produce either tall or short plants, depending on which allele are passed down from each parent through meiosis.
It does not matter whether both parents are tall or both parents are short. Both types follow genetic laws such as the law of segregation, which deals with gene separation during gamete formation, and independent assortment, which deals with how genes are passed down independently from each other without being influenced by other genes.
So to review, monohybrid crosses involve crossing two parents with different versions for just one trait, while dihybrid crosses involve crossing two parents with different versions for multiple traits.